uh, 7,000. I think Indonesia has around 10,000. So it's within those range. How many endemic species? I actually don't know. So let's talk about the importance of Lepidoptera. So very simple. Um, what's this? Illustration on butterfly, predator, and plant interaction. As you can see, you have butterflies and moths, and then usually most of them or some of them will have habitat preferences. So various habitat types, there will be species that can only be found there, and there will be species that can be found in all habitat types. And of course, butterflies and moths, they feed on flowers, getting nectar sources, which actually um, contributes to pollination. So butterflies are actually noted for their pollination services. And some moths are actually they feed on blood, so especially the vampire moths. So they feed on mammals. So butterflies and moths, they have larval stages. And larval stages are very important because some of them they feed on certain hosts. Some species are host specific. They only feed on one host plant. And some of them actually feed on various hosts like grass, Muraceae, Fabaceae, Melastomataceae. So different plants will have different types of caterpillars. And some hosts are not just plants. Some largest larval stages actually feed on small insects like um, mealybugs. So in terms of predators, they're also very important, especially with birds, bats, amphibians, and reptiles, because of course, butterflies and moths and their larval stages are food sources. So this is just a simple illustration on their interaction. Why butterflies and moths are actually important because of this interaction. So studying basic ecology of lepidopterans, let's start with butterflies. So when you go into the mountains, you always look for the best site. Forest trails, meadows with flowers, and of course, forest edges. And this is one of I like this kind of site. Whenever I go into the field, whenever I see this landscape, this is in Amlan, Negros Oriental, and that's Mount Ginsayawan at the back. So whenever I see very thick forest on each side, there's like a river, and there's like a place like a short trail or something to walk around. That's actually a good spot. And a lot of people who study butterflies, when they see this, they would say, I'll stay here. For me, I would say I can stay the whole day in this place catching butterflies. The best time to catch butterflies is actually morning. They peak at around 8 to 11 a.m. So that's your peak hours. Beyond that, you will probably see one or two butterflies flying around, and then once you reach dusk, of course, moths will start to appear. Always look for a best site: forest trails, meadows, and forest edges. Example like this. So this is in Apayao, North Luzon. We were there in April, just uh last month actually. So again, this is a good forest, and we just look for again a best site, a forest trail, a river bank, and that's going to be the best spot for you to study butterflies. So this is in Apayao last year. I went there last year and also this year. So that's me in the middle with an insect net, and we have the guides and some researchers. So this is an example of a forest edge. So there's actually a river on the leftmost side. You can see some there, that's the water. So we usually just stay there, observe. This was probably around 9 a.m. So we stay there and collected butterflies. Again, choosing the site is the best way to study butterflies. Here's another one, a forest trail in Apayao. As you can see, you have forest on both sides, and then you have this clear forest trail. Yeah, we usually just walk around, and then we were able to find a lot of butterflies. That's because they like to fly on open spaces to bask under the sun. And of course, they like to display their colors and patterns where the female can actually see them. So they usually perch on these bushes on this side. So if you see a trail like this, just walk slowly and try to observe what you can, because once you see the site, chances are butterflies are already there. So you just need to walk slowly. If you have keen eyes, I would just look for the leaf tips because they're usually there. So again, this is me in Mount Talinis in 2022. Aside from taking photographs or collecting butterflies, it depends on the landscape if there are no butterflies, because I think the drizzling. So as you can see with my raincoat, it was drizzling. So instead of looking for butterflies, chasing butterflies, I usually notes. So I took notes of the habitat and what are the things that I saw flying. So it's also very important that we still need to take traditional notes. We had phones, but you know, phones, when they lose signal, I mean, when you lose battery, I mean, that's the end. Notes anymore. This was me in Mount Tapulao. This is probably I was there for like 7 times. You've been to Mount Tapulao. I mean, it's a very nice mountain to climb. Um, going, I have an explanation on why I always go there later on this slide. So this is Mount Tapulao. It has forests at the higher elevations. It has a very nice lowland to mountain forest. I started going there in January of 2025, and the photo was taken March, 3 months ago. I was there 3 months ago. So this is a good site to study high elevation butterflies. You have the forest trail, an open field, and you will just see butterflies flying back and forth along the trail. So that's why I like this spot, because I don't have them, because they usually just fly along the trail. I will just have to choose a spot and then wait. And then of course I along the trail, and then you some interesting butterflies like this blue Euplea mulciber, Iptima sempera on this one, and then have the Discolampa ithion, and then a Arophala, this brown butterfly. So if I don't like collecting, I usually like to take photographs, especially for, you know, publication purposes, and also for something doing like this, presentations. So and then I was in um Apayao, and depending on the season, when I was there in April, there wasn't that much butterflies, but that's because most of them are still in their larval stages in April. So when I went there, I went out, tried to look for butterflies. There wasn't that much butterflies. So I said, okay, it's me that they haven't emerged yet. So I started looking around the forest trail, and there they are. They are still at the larval stages. So remember, caterpillars and host plants. So whenever I go out, I usually ask in the group, who studies botany? Who studies plants? And somebody said, I do, sir. And I said, okay, I might need your help later on. So I went out and look at the caterpillars. And this is a uh, caterpillar. I check the plants, uh, ask for an ID, and it's on feeding on a Phyllanthacea. This caterpillar is very unique because it makes this. So that's the frass, and then it's has this trail. It usually does this to prevent being harassed by ants, because if the ant try to eat it, this caterpillar will just retreat towards here. The ants cannot travel here. So that's why it's in defense behavior. Okay, here's another one. Actually, this is the second larval instar. This is the late larval instar of that caterpillar, feeding on Phyllanthacea. I found them on the same plant. So this one is to pupate in the next few days. So again, it will emerge and we will ID the adult. So if there are no butterflies, it's also important that you can actually check their caterpillars, because a lot of the life histories, the larval butterflies here in the Philippines are still unknown. Out of 900 species, probably we only documented around 300 to 400. The rest are still unknown. We don't know how they look like. So if you find one, try to rewrite, and then let's see. Maybe you will make some discoveries on the life stages of these butterflies and moths. That's uh, the uh, the larval stage is actually diagnostic, and especially or taxonomic, and also the head capsule. So depending on how the spots are arranged, the white spots, different species will have different marking. Okay. Again, this one's feeding on Phyllanthacea. This one is feeding on Medinilia. So this is a moth caterpillar, and if you see something like that, like gummy worm, something that's very colorful with, with hair. Tendency is they have urticating hair. So handle it with care if possible. Do not touch it. So this one's feeding on Medinilia. So if you have a Medinilia on your site, look for this. They feed on the flower and the flowers. This is a moth, possibly in the genus Calizigena. So, bearing one of these. And this one is feeding on Vaccinium. Is a caterpillar that we actually don't know what it is. So we're waiting. Actually, the caterpillar is now on its pupal stage. So it's on its cocoon, actually waiting until it emerges. We already have what's this? Initial ID on what it could be. So again, if you want to document something new, chances are rearing caterpillars, definite chances of something that's new, it's gonna be like 100%. So we're waiting for it to emerge. So again, it looks like a gummy worm with the eating hairs. Handle it with care, or else it's gonna probably be painful. We haven't tried. We don't want you to do that kind of. Okay, and this one was feeding on Vaccinium. There's another caterpillar that was feeding on Vaccinium. This caterpillar, as you can see, it has many thorns in its body, and it makes this fast trail. It makes trail so that the birds will be confused which one is the caterpillar. It's either this one or this one. So whenever this caterpillar moves, it actually carries this its frass. And it actually makes this frass trail as an escape from ants. So there's frass trail here, frass trail on the edge of the leaves. So again, it's another, whenever I go into the field, I usually look for frass trails, the tip of the leaves that's like half eat. Chances are you will see butterfly at the base or at the tip of that leaf. Okay. I go out in the field. I carry small sauce Tupperwares or uh what's this? Ziplocs. So I take them out and then go into our campsite and then take pictures, study it, examine it, and then feed it until they reach pupal stages. Once it reaches, that means it's going to emerge soon. Chances are, if you collect a very small caterpillar, you yes, it's very picky with food and environmental conditions. Sometimes, sometimes you have to rear them in AC, especially if those butterflies are actually found in high elevation where it has cooler environments. Chances on lowland areas, they sometimes don't like the heat because habitat preferences. Hey, how about moths? So again, it's look for the best site. Various habitats will have you have different results. Again, forest trails because moths and overlooking our areas if you're going to set up a light. And if you're going to set up a light, it would be best if you're going to set up light moonless nights. There is an explanation on why it has to actually because of the moth's orientation always or attraction towards the moon. I actually wrote a theoretical paper on this one. The moon is tends to become the greater source of light. So when you set up your own light, chances are the moth's gonna not go somewhere else because it's about their orientation and maybe traveling using the moon as their calibration. Okay, so when we set up our light, we usually use Lepid because this one has a white light and a UV light. So you have all this spectrum which all the moths will be attracted. So using light to attract Lepid light. Maybe your house, if it will have a strong light, especially white light, you will probably get a lot of moths. In Apayao. So as you can see, it's overlooked. We set up our light trap on this small, and then that's it. This is early evening, 7 p.m. Not that much moths yet, but at around, so this is our site overlook, and the light trap is here. So this was now around 8 p.m. A lot of moths. Maybe you can see something like this. So this was around 8 to fireplace, oh? 8 to 9 p.m. After 8 to 9 p.m., there's still a lot of moths, but the thing is that it's gonna be all same species. So what we did, we set up the first night, and then the second night, and then because we already collected it species. It's gonna be the same. You'll be collecting the same. Maybe there's gonna be another five, but the problem is we have to go back after the specimens and what's this? Uh, Okay. Okay, so again, if you don't want to collect, you can also go around the light traps. Some of them will actually just be on the bushes or maybe just looking at the light somewhere around the light. So I took the advantage of that to take photos of those moths and some moths along the trail. You will get a lot of Uh, some of them I actually don't know, and some of them are actually first time these kinds of moths. So as you can see, the diversity of moths here in the Philippines is still undocumented. You want to do, especially in systematics or taxonomy, new species, moths would be there was a student in Ateneo na working on a family of moths, Musitimini. So maybe you guys, some of you, can actually study different subfamilies of moths and contribute to the diversity um possible 10,000 species that we have. So current research in the Philippines. So I was in Mount Tapulao uh for many months, not many months, many times. So what we did was doing the butterflies in Mount Tapulao, which probably you can do this in your area, especially in UP Visayas, in Mount Majaas, especially in Panay. We describe a butterfly there, Ticandra negrucensis Angel alcali, named after Alcala. It's a endemic butterfly, and that subspecies can only be found in Mount Majas. So here's in altitudinal distribution. So we river and then to Flores Claveria and then towards the high peak. So we recorded around 40 species of butterflies. There could be more, but one of the highlights is this one. So if you go to Mount Tapulao, it's actually a gradual ascent, and then you will have those rocky ascent somewhere mid elevation, especially at 1,200. So different elevations, you will actually get different types, sometimes different types or different kinds of species. Examples this one. So we started climbing Tapulao early morning, and I was just walking slowly and try to observe what butterflies I can find. In lowland areas, they're mostly grass specialists. So I was like, okay, let's go. And then once you go into the mountain forest at elevation 1,200, we found this butterfly, Delias georgina. I walked again, and then I found it in our campsite again at 1,700 meters above sea level. So this butterfly is actually found at higher elevations. And then it was first found in Baguio at around 1,4 to 1,5. So that's its probably elevation. And then I was with a team of botanists, and I said, this butterfly feeds on this specific plant, only to find out that starting that elevation of 1,200, that host plant, which is a parasitic plant, was actually found. So I actually went to the plant only to find out that I didn't know about plants. This plant is parasitic and actually grows on tree tops. I was trying to look for eggs and caterpillars, and I said, "There's no way I'm gonna climb that." But we found the host plant, and the botanist team were able to collect the plants. I checked for eggs. There's no eggs, so they're probably not laying eggs at this time. So we found it here. Since the butterfly is also distributed at 1,7, there's a possibility that the distribution of this host plant is actually so. Remember, species of butterflies are they're sometimes very specific to their host plant. And then we have this another paper, and then only to find out that we found the pupa in Apayao, and we found the pupa at around 760 meters above sea level. So, and then we found that below this possible host plant. Helixanthera, another parasitic plant. So it was thought that this butterfly is only at above 1,000 elevation. But this in Apayao, we found them at 760. Now, the question is this a recent elevational shift or dependent on host plant distribution? So there's going to be, this is a good research, especially with what we're experiencing now with heat index. How do you think this high elevation species are going to behave? And especially those at the middle elevation. Are they gonna go up? Are they gonna stay there? Or do you think they're gonna go somewhere else or stay at lower elevation? So we don't know what's gonna happen, but so far this is the data that we have. Okay, so another thing that you can work with is if you have urban parks in your cities, you can actually work on the butterflies there. So this is our paper on the assemblages of frugivorous butterflies in two urban parks in Quezon City, Philippines. And another one is this paper that came out, University Campuses as Urban Biodiversity Refugia, especially for butterfly diversity. I mean, UP Visayas, maybe you can also do or those who are listening here, if you want to do your thesis on butterflies in your campus, that's a good start. Sometimes campuses have very uh, oh, elaborate gardening system. So it will actually attract a lot of butterflies. But again, it depends on the the weather or the kind of habitat that you have. Chances are, if your campus is near a forested areas, you will definitely have some interesting species. So, in terms of butterfly ecology, we need to understand that there's one disturbance that rules them all: typhoons. I mean, when typhoon comes in, it's like it affects everything. So I'm gonna show you a brief what we did with the ecology of butterflies with typhoons here in the Philippines. This is my dissertation. So here are typhoon records from 1951 to 2003. So as you can see, we get hit by a lot of typhoons, 20 plus per year, and they're getting stronger and stronger. So I had field sites on Negros, North, Central, and South, but let's focus on the North to shorten everything. So in North Urban, this is North Urban, this is Bacolod City. The suburban, it's like the sugarcane plantation. And then you have the North Forest. So in urban, it's usually the University of St. LaSalle campus. And here we have a sugarcane plantation, and we have the forest here in North Negros. So we compared typhoon and non-typhoon. Oh, here are my field assistants from University of St. LaSalle who participated in data gathering. So we found out that if you want to study the ecology of butterfly, sometimes we need to get the right timing. So here are the species here in the abundance. And as you can see, they became more abundant during the intertropical convergence zone. This is the first rains of June. The first rains of June, after that, we saw a lot of like the emergence, abundance of butterfly species. So that's a good um timing. Well, if you want to compare dry and wet season, you can now you can compare. So there were significant typhoons that passed by my sites. You have Lawin, and then you have Karen, and then you have Marce, which actually went directly to our sites in Negros, and this one, Nina. And then only to find out that again, typhoons, the first abundance here is the intertropical convergence zone. And then you have the second is Typhoons Lawin and Karen, Tropical Depression Marce, Typhoon Nina, and the rest were actually those typhoons that actually caused rains in our country. So as you can see, we still have the the wet and dry season of butterflies. During the dry season, there's not that much butterflies. It's during the wet season that they will be abundant because during the wet season, there's rain. That's a signal for also for plants to maybe to have fresh shoots and also bloom flowers. So that means butterflies would have to have timing for that one. So in terms of species diversity, as you can see, from forest to urban, it's actually getting lower. North forest, yellow, north suburban, green, pink is the north urban. Same pattern with central, same pattern with north. So in terms of typhoon season and not non-typhoon season, it's actually the same diversity. It's almost the same. Instead aside from the urban, they actually, I mean, urban is kind of the unstable type of environment. Things are changing. So species sometimes keep on changing. So in terms of species communities, urban and suburban did not show any significant difference. That's because when you modify the habitat, chances are those plants that there the species of butterflies is going to be feeding that plant is going to be the only butterflies that will be there. So if you can see the non-typhoon and typhoon season is almost the same, except for forests. There's a significant difference during the non-typhoon and typhoon season. During typhoon season, a lot of species that are actually emerging during the wet season are still there. So they still have the dry and wet season assemblages. Okay, so applications in agriculture, very short lecture on this one. It might influence the distribution and population of agriculturally important insect pests. So again, you will have, this is very important in our country to have database in insect distribution because this is going to improve our integrated pest management by predicting insect movement and population during typhoon season. For example, you have different plantations from different areas. Typhoons will pass by, but you have pests on this area. Then you have typhoon. What are the chances that some of these insect pests will be transferred to different agricultural areas of the country? If we have a database of these insects, especially with the caterpillars and moths and other insects, then maybe we can have a nationwide database. There's a typhoon, and actually, it can actually improve our IPM by forecasting insect movements and distribution to different crops or islands in the Philippines. Right. So, talking about typhoons, this paper was published in 2004. Not 2004, 24, 2 years ago. This Philippine butterfly actually went or was observed in Hong Kong. And when we noticed that there were two typhoons active in the Philippines in June 2023, and the butterfly was observed in July 5, 2023. So this is endemic Philippine butterfly, possibly brought to the to Hong Kong through typhoons. And it's not just one, because this caterpillar was photographed by our citizen scientist Linda Alisto. This was photographed in Batanes. And this caterpillar shouldn't be there because this caterpillar, the species found in Yaeyama Islands, especially in Ishigaki, on the easternmost of Taiwan. How do you think it got there? I mean, the caterpillar did not swim. Possibly that the adult was able to reach Batanes. The only possible is typhoon. And then the adult was able to find its host plant in Batanes, laid eggs, and then that's how the caterpillar probably will emerge. So we might do field work there because we were talking with my colleagues in Taiwan, and he said that it's true that some moths and butterflies from the Philippines actually reach the southern port of Taiwan after typhoons. And those moths and caterpillars and those moths and butterflies will actually just die there in Taiwan. That's because they don't have their host plants. So host plants is actually a limiting factor. Okay, so reviving an extinct color. So this butterfly is considered extinct in Cebu. This butterfly can also be found in Panay, but it hasn't been documented in Panay for many years. Knowledge of its ecology, biology, and life history. So if you find this butterfly in Panay, take a photo, and that would be nice. So this butterfly is considered extinct in, and then last year, another citizen scientist, Romana de los Reyes, was taking photographs of butterflies in her garden. He photographed this, uploaded, and I couldn't believe I saw it. I emailed her, messaged her, and said, "Can you rear the eggs? Because this is a female, and it's actually laying eggs. As you can see, it's bending its abdomen." Okay, so we were able to rear it, the caterpillar, the pupa, and so, okay, why is it important to study the life history of this butterfly or all the butterflies? So, long life history probably prone to external threats such as typhoon. So we documented this life history of this butterfly. The eggs will last 3 days, larval stages 16 days, pupal 7 days after emergence. Life history of swineson, pupal stage 6 to 7 days. And then the other one, pupal stage 5 days, 5 to 7 days. So it's usually pupal stage is within 5 to 7 days. Now let's take a look at this extinct butterfly in Cebu that was found in Negros recently. The egg, it lasted days. It emerged, the first larval instar, lasted 2 days, second 3 days, third 5 days, fourth 3 days, fifth 4 days, pre-pupa 2 days. Now look at the pupa stage, 135 to 162 days. The ones that we reared is usually 5 to 6 days. Now the pupal stage of this one is 135 to 1. That's not good. So, and their host plant is actually not rare. Their host plant is pomelo. So if you have pomelo in your backyard, uh, if you find an interesting caterpillar that they look like cucumber or something, or they laid eggs on June, served it. So maybe next month, the first rain, if you have pomelo, if you have time, just look if you find this butterfly laying eggs. So, so it takes 2 to 4 weeks to complete the life history. But the Papilio hipponos took 135 to 162 days. That's like 5 to 6 months to complete its life history. That's probably the second longest of all the butterflies. The first is the one in Colorado. It was 2 years. This one, 5 to 6 months. So shorter life history may allow the species to avoid external factors such as typhoons, while longer life histories are probably prone to it. Adults usually synchronously or within a window of few days to mate and lay eggs. And yet the hipponos emerge 1 month. 1 month yung pagitan nila. So that means the male will have to wait a month for the female to come out. And a female butterfly can usually lay 200 to 400 eggs. And in our observation, we only obtained 4. One Japanese scientist in 1974 was obtained an egg from Bohol, and it he was able to obtain an egg, and the pupal stage lasted 3 months. So if you find that one in Panay, let's see if it will probably have a shorter month or a longer month of. So why it's not good? The eggs were laid in June 10, it pupated in June 26. And then in August there were typhoons during this week. In September, in October, and in November, Ma'am Liana actually used a different cell phone. That's because a typhoon hit in northern Negros and toppled trees and electric poles. That means she didn't have electricity. She has to use data from her student, and then the male emerged in 7. And then there's another typhoon and another typhoon in December. The female emerged here. Now remember, we reared them indoors. What if that caterpillar or pupa, what if the pupa was just left outside on its host plant and the trees and the strong winds around her garden, I mean, the trees were toppled down because of strong winds. So what are the chances that they will not survive there? So we were able to rear them. We released them in the wild, and hopefully it will come back this June. So that's the hope. So it was also last year in June that the tropicals were everywhere in the news. That's it's probably the long dry season, and then probably weird dry season last year. And then it rains of May and then June. They just went into emergence. Here, this was also in June 15 when butterfly migration in Palawan. That's because after the first few heavy rains. So last year in June, lots of tropical moth swallowtail emergence. You have the migration of the Euclea butterfly. And then you also have the extinct butterfly that was documented in Negros for the first time. So reviving extinct color, that's Ma'am Romana. So taking a photograph, that's a female that just emerged. Now it's actually published. It's open. Go to Google, type this title. You can, if you want to look for this butterfly in Panay, it's there, but it hasn't uh maybe for 20 years already. So if you want to look for it, you can download the paper. And then of course, we're not testing the plans and methods to revive extinct butterfly. We're dealing with genetics, ecology, and physiology. So have the pupal cases of those butterflies, and we're going to extract DNA, hopefully successful. Next month, we're going to do DNA analysis on that one, and then let's see if we can find more of them in the wild and study again their ecology, possibly their physiology, on why they have such long pupal stages. And ecology, do you think they have preferred habitats? Where do they go? Where do they spend most of their time? So many questions to ask. And acknowledgements, I'd like to acknowledge the late Colin G. Treadaway. Um, I uh, I learned a lot from him. He's the top authority of Philippine butterflies. Still, I have some of his emails and correspondence on what on butterflies. And of course, with Justin Uida, this was in his house, and we're just at his collection and looking at literature and notes and studying biogeography and distribution of butterflies. I'm actually going back to their house to check on the collection. And of course, Dr. with field of conservation with mammals and doing that. And of course, with my advisor, Dr. Thomas C. Emil. I work now on typhoons and butterflies, and he did actually work on hurricanes and typhoons in Florida in 1992. Again, on how the typhoon and hurricane affected the shell's swallowtail almost into the extinction, and they were able to recover. Nation. All right, I think that's it. I am happy to what's this? Uh, there are any questions? Happy to what's this? Answer.
Ayan. So, thank you so much, Sir Ja. By the way, Sir Ja is educated from Silliman University for his bachelor's degree. So, yung mga taga Negros dyan, ayan, ay taal na ta Negrense yan si Sir Ja.
Yeah.
And educated from, his master's and PhD is from the University of Florida. Amerikano yan. Napansin nyo may accent siya? Amerikano kasi talaga yan si Sir Ja. Hindi siya hindi siya Pinoy. Amerikano yan eh.
Na pag
Yan.
lecture I have accent pero pag ano lang local I have my other accent.
Oo, nagbabago yung accent niya depende sa kausap niya.
Yeah.
Ayan. So sige. So, I think our participants now can ask already their question. So if you have a question for Sir Ja, kindly raise your hand so I can address you. Kindly say your name and the institution that you belong to so that you can address your questions to Sir Ja. Anyone who would like to ask a question? Naku, yung mga estudyante ko, pag walang tanong, alam nyo na, tres ang grade nyo. Ayan, may nagtaas naman na I think we have one. Sige, so, sige, unahin natin si Carl. Yes, Carl? I think you can allow him to unmute. Ayan, sige. Ayan.
Hello, sir. Good morning.
Mhm.
Audible ako, sir.
Yes,
Yes,
yes.
Well,
yes.
thank you. So, sir, I am from the Visayas State University in Baybay City, Leyte,
Mhm.
and I am an undergraduate. I'm still having my undergraduate of environmental science, and um, uh, my study is about arthropods, no? Comparative comparative assessment of arthropods and stratified microhabitats. So I am already um, finished in my ID, and I am still uh, in struggling in how to paano siya i-start sa yung sequence about sa analysis ng ang aking study. For example, yung mga abundance, mga ganun. Especially, my ID is only up to family level lang kasi mahirap kasi yung species level.
Yeah,
Yung
yeah.
mga ganyan, sir. So,
So you want to know how to start.
yes, sir, especially with the analysis in interpretation of the data, like that, sir.
Okay. So looks like yeah, how many sites do you have? So
Uh, it's about like 5 sites, sir.
okay.
Understory, forest floor, riverbank, forest edges, and low canopy, sir.
Okay. And then how many times did you sample those sites?
I sampled it every day
Day.
for about, I think it was about uh, 9 days.
Okay.
Lahat-lahat. Oo.
Okay. So 9 days each, something like that.
Yeah, something like that.
Okay. I think with that you can actually analyze again the usual ANOVA, the usual ANOVA per per habitat, and then you can actually do non-metric multidimensional scaling that it will actually analyze the similarity or dissimilarity of your communities. So it will actually show you if yung canopy is gonna be similar with the other canopies or not, and then it will actually do that for the kind of data that you have. Yeah.
Okay, sir. Thank you, sir.
Ayan. So before I ask another one, siguro additional advice kay Carl, tignan mo uli yung iyong objectives because that might help you in figuring out how you can start with your analysis. Because all of our what we do in research usually nakabase yan sa ating
Objectives,
objective.
yeah.
Okay. So
Okay, sir. Thank
let
you.
me call on Steven, no, to ask his question.
Hi, sir. Good morning po. Marinig niyo po ako, sir.
Yes, yes, yes.
Yes po.
I can hear you.
Again po, good morning, maayong agaw sa tuntanan. I'm Steven John Meliandas, a research assistant from Sultan Kudarat State University, province of Sultan Kudarat. And thank you for this opportunity po, Sir Eric and Sir Jade, for this wonderful. Actually, I'm very interested in this one because my undergraduate study po na natapos sa I graduated last 2024 is more on species diversity, particularly species diversity, but in mushrooms po. But I have a lot to learn something about butterflies, Lepidopteran, and other kwan po, sir, kasi very interesting pong topic na ito, especially sa ating mga biology student that under in the program of tawag dito sa advanced ecology nila na stage. Actually, sir, I have a parang tatong question, sir. Dalawang parang concept, parang circuit of parang pang national talaga na level of research question, and your parang personal opinion, sir, personal experience. For the first question, sir, yun nga, yung sinabi ko po kanina na this webinar is intended for the students of BS Biology. So what is your advice to those students, sir? Particularly yung na-mention kanina ni Carl. Before I forget, madagdag ko na rin kay Carl na aside subjectives, you must revisit the published articles kasi doon mo yes po, doon mo makikita kung ano talaga yung need mo, particularly sa methods na bubuin mo po. So, I advise you na sa methods nyo po, you must to visit published articles related to the study po. So, yun po, sir. What is your advice to the students to be more interested in studying Lepidopterans, and what is your parang personal stand po, sir, na these observations must be boost, particularly in the species diversity of butterflies. Can this study, sir, especially yung sa mga bata, sir, pwede po ba natin ito ma-present sa DN or to have a parang TWG approach po, sir, na yung base kasi I also attended one webinar past 3 weeks ago na ata, sir, if I'm not mistaken, that it stated that yung lahat po ng mga results ng mga bata po, sir, is kailangan i-incorporate or parang gagawa ng parang isang recommendation paper and ipapasa doon sa kwan po, sir. Parang ang naiisip ko rin po, sir, that it is high time to the Department of Environment and Natural Resources that this kind of species diversity study is panahon na kailangan tubos kasi yung second question ko, sir, is you mentioned, sir, kanina na 900 plus lang ang species ng butterfly here in the Philippines. So what are ano po yung possible factors po, sir, na kailangan na baguhin ng DNR, particularly in the species diversity po sa ating butterflies? Sorry po, Sir Erica, medyo na ano po. And for the third question, sir, you presented also earlier, sir, your experience sa mga sites mo, sir, in Apayao, in the Negros Island regions, sir, so and so forth. So, what is the challenges you encountered, sir, while you on the ground sighting the butterflies, especially sa hapon hanggang paggabi na, sir? Kasi we all know pagbukid doon po na uuna na mag umuulan kung minsan. So, ano po yung mga factors, sir? Ano po yung kailangan paghandaan, and also to the students na kailangan when you see you conduct on the ground, kailangan 101% kailangan nyo pong paghandaan ito. Yun po, sir, and good morning po. Sorry po, medyo napadami po. Thank you po.
Yes, I see.
Medyo intense yung question ni Steven, pero I think kaya
Yeah.
naman sagutin niya ni Sir Ja.
Yeah.
Sige, Sir Ja.
There were three major questions. So the first is how to actually convince students to study butterflies and moths. So what I noticed is that a lot of students would usually engage into this topic when you show interest. An example is I have a lot of students actually emailing me for ID. And then of course I would reply and said I can ID this within this week or next week, or this will take long. And then I'm going to show them that some of these species, or let them know that some of these species are actually, because some of them are actually collecting different sites. So I would usually tell them that it would be nice if you can write a certain article of this one with the species and the species list, and then we can publish it to example short notes, which is highly um, recommended because it's not very technical, but it's straightforward telling you the data that you have. So that's one. Make students interested in this topic is actually for your mentor or your teacher to also be excited about the topic. And all of you will be excited. That's what happens when I did my undergrad thesis. So my undergrad students usually work on this butterflies, and I usually go with them in the field and then, show optimism on data gathering. So I think that's one. And then next is with the DNR. I think maybe what's this? I um I think the question, what was the question about DNR? About insects? I'm not sure if I mean they should have also have maybe a different support with studies of insects, because as far as I know, we're sometimes most insects are considered pests, and this is why we don't. The insects are least appreciated because of that factor, but none of them aren't. So as much as possible, when we have results in our expeditions, when we have results from our research, we usually give copies of those results to DNR. So that's what we usually do. And before we do field work, we usually propose it to the DNR. And then after many months of sorting the data, it's either we present our results to the DNR or we will give them our report. So that's what we did with Mount Tapulao. We went to Mount Tapulao, gather data. Once we went down, after a few months of sorting the data, identifying, we actually gave them data. Recently, we went there, we gave them posters of the plants, fungi, and birds that we found in Mount Tapulao. Of course, I'm gonna be giving them butterflies and moths and other group that was working on insects. So it's actually the importance of data availability and data sharing. So I think that's important. So whenever there are new discoveries or some new species or new locality record, usually with Facebook, it's gonna be there. So it's up to us how we're going to share it. So your last question is difficulties in the field. Yes, we have so many difficulties in the field, and I don't know which one to start because the first time I did my field work was in 2012, and ever since I've been doing a lot of field work, maybe sometimes 5 times a year. So difficulty is of course, you're restricted to your budget. You're restricted to your budget. That's the first thing. Second is logistics. Who's gonna be there? Who's gonna be with you? And how are you gonna survive there? Remember, when you go into the field, you have all the budget. When you go into the field, remember, there's no 7-Eleven in the middle of the mountains. So everything has to be prepared. The food, especially food. You need to have a food that will last you. In our case, we usually need a food that will last 5 days. After that, we have to go down. And then next is where to camp. It's very important that when we set up campground, you just don't go on Google Maps and say, okay, I'm gonna camp here because it's in the middle of nowhere and species gonna be interesting there. We don't. We usually um camp first when there is a water source. So if there's a waterfall or a river, chances are we need to camp there. We need the water source. Next is you need to ask, of course, you need to do a courtesy call of the LGUs. And then so that for your safety, because the LGUs will know the status of everything in that area. So we usually do courtesy calls of the LGU, tell them about our plans, our research, and then they will tell you where to camp and who's gonna be your guides. So in terms of the weather, yes, there are times when we go up in the field and then we didn't know, only to find out that when we went down, there was actually a typhoon. There's no Wi-Fi. So it's like strong winds and rain. And then we went down, it's like there was a strong typhoon. And like, really? We didn't know. We were in the middle of nowhere, there's no Wi-Fi. So I guess this is what I would usually say. Field work has a lot of unexpected events. Either you go there, collect nothing. You go there, you will have all the species that you want. You go there, it's either things gonna be perfect. You go there, things it's not gonna be perfect. So when you go field work, it's actually you need to what's this? Expect the unexpected and don't expect too much. As much as possible, when we do field work, the most important thing that we do in field work is we enjoy. That's it. We enjoy, we learn. We go there, we collect data, and then enjoy what we have. And then if you're not satisfied, we always say, okay, it's not a good sampling season. Let's look for another dates. This is why other researchers, they just don't go to one site once. They go to one site and then they visit it multiple times. So it's actually look for the best spot. That's it. And it's always a trial and error. It's either you get it the first time or you're not gonna get it. You're only gonna get it after many times. So I hope that answered your
Yeah, I think. Ayan.
questions. Yeah,
Ayan. Thank you, Sir Ja. Thank you, Steven. By the way, no, to our young scientists and of course our environmentalists here, no, as much as again I understand the clamor of the weaknesses and of course the sometimes we think that DNR lacks a lot. However, it's really on the communication side that we need to communicate with them. We need not to always feel that we know everything because there might be some things that we don't understand in how things work in the DNR as well. So we really just need to communicate with them. And again, as much as we are in the academy, sometimes you feel that we know everything, but we don't. No, so we really need that communication. Kasi nakakalimutan siya minsan ng mga environmentalists, ng mga students, no, that we should not always be an antagonistic sa kanila, sa mga government institution. Kasi hindi rin natin alam kung ano yung kanilang mga challenges. Sige, before I call in another audience, there are some questions here in the chat box and I'll compress this one. This is an interesting thing, no? Sir Ja, their question is actually on preserving butterflies and moth specimens. So what is the best way to preserve butterfly and moth specimens in general?
Okay, to preserve butterflies, and actually, I find them difficult. That's because of our humid environment here. So actually, if you collect insects, and then with the humidity, it will just be gone in the next few days. So here's what I would usually do. When you collect butterflies and moths, of course, with the necessary permits, when you collect them, I usually put them in wax papers, envelope. You make triangles or envelopes from wax papers. And then here's here are some of the options that you can do. You have those envelopes, and then I usually put them in the freezer. So put them in Ziplocs, put them on Ziplocs or Tupperware that's sealed, and then put them on the freezer for many that would last for many months. And then with what's this? The other option, if you don't have freezer, you have butterflies and moths, put them on wax paper, triangle or envelope. You put them on Tupperware. Alam mo yung Tupperware na may rubber seal? That's a good one because it will maintain its humidity inside, and it actually prevents other pests from eating it, mga dermestids or other ones. So that's an option unless you have a very laboratory that's sealed and with AC 24/7. It's very difficult to preserve here because again, it's the humidity. That's very number one enemy of insect collection. Yeah.
Thank you, Sir Ja. Another question from the Q&A box. Here's one from Kyla from Mindanao State University General Santos. So she will be conducting her undergraduate thesis involving Lepidopteran diversity and geometric and morphometric analysis. So she would like to ask, during field sampling, what are the usual techniques or characteristics that you use to distinguish male and female Lepidopterans?
Okay, so in the field, to determine whether a male and a female Lepidopterans, actually there are many ways. First is some of them the male and the female is gonna be different. So before you go to the field, download a lot of literature on the family, your target family, especially geometrids and sphingids. They will be there's going to be a lot of literature on that one. And then look at the male and the female. Morphologically, the male is actually smaller than the female. And then the other one is, aside from the pattern, male and female will tend to have different patterns, and in some cases same. If same patterns, look for the smaller ones. That's the male, or and then the female is going to be larger. The other one is when you collect it, look at the abdomen. If the abdomen is usually small, short, stout, and pointy at the tip, that's usually a male. If it's circular and then large and then at the tip is flat, that's because the tip has to be flat because of laying eggs. So that means that's a female. So I hope that's and then I mean it will take a while for you to actually notice it, but once you collect it, you should try to examine it. If it's pointy, it's a male. If it's flat, that's going to be a female.
Thank you, Sir Ja. So before I move again to the question and I'll call on another participant. Adiel?
Hello po, good morning. Rinig po ako.
Yes, yes.
Yes.
Sige, I'm Adel Balanza po from Cavite State University Indang Campus. So, may undergraduate thesis po kami about sa Lepidoptera diversity po sa aming lugar, saka yung kanilang potential parasitoids po. So, isa po sa mga plano namin ay mag-force copulate nga po ng mga Lepidopteras na makokolekta namin sa field. Tanong ko lang po kung possible po ba yun sa lahat ng species kasi alam ko may mga iba na kaya ho yun at kung kaya silang paitlogin po kahit wala po sa kanilang host plants po. Yun.
Okay, the question, you're studying parasitoids with
Yes po.
Lepidoptera and then gusto nyo ng mag-rear
Yes
na,
po.
and then ang difficulty nyo is yung anong difficulty nun?
Yung kung kaya pong i-force copulate po, force breeding
Yes.
po.
Depende sa species. Some, ano yan, moth or butterfly?
Both po ang aming po.
Yeah, try hand pairing them. Usually, hindi mo yan, sometimes hindi sila magko-copulate the first time. So sometimes you just need to do it slowly. You need to have a lot of males and a lot of females because some of them will just not copulate. So it's actually it takes a lot of trial and errors in order for them to copulate. And then of course, you just need the host plant. The other one is if they don't copulate by hand pairing, you can buy yung mga collapsible na something sa Shopee that may mga zipper, collapsible mesh net or containers, and then put the host plant, put the males and the females, and then hopefully they'll do their job in mating on their own and also laying eggs. So that's another option.
Yeah. So I hope nasagot ni Sir Jaa yung question ni Adiel. So I think we can call on Mr. Alex to ask his question personally to our famous lepidopterologist.
Sige.
Morning po. Rinig po ako?
Yeah.
Good morning po sa lahat. I'm Alex po. From UPLB naman po ako and forestry undergrad po. Since forest entomology po ang specialization ko and give lang po ng context na nag-internship po ako sa Marinduque kung saan po ay parang hotspot for butterfly farming. Ang question ko po, sir, is do you think po or parang napapansin ko po kasi parang there's a lack of study po when it comes to genetic biodiversity, lalo na po sa captive breeding ng butterflies. Since parang one of the regulations sa butterfly farming sa Marinduque is they reproduce and release ng 10% of the population. Do you think po may underlying or parang do you think po na yung genetic diversity could affect long-term po sa pagbi-breed at pagre-release sa total population po ng butterfly population sa Marinduque? Ayun po.
That's an interesting question. There are no current research on how it actually affects its genetic diversity. Because if you take a look at the reproductive biology of butterflies and moths, they can lay eggs 200 to 400. So that's gonna be a lot. So when they breed it and then they rear it and some of the parental stock's gonna be there, as long as probably they're just releasing it into the wild. I think kasi that's already a mandate for them when they breed. Some of the 10% is gonna be released into the wild for again to maintain the diversity, the genetic diversity. But with the breeding, I am not sure because insects are capable. If you breed insects, I mean you can do that. But chances are the genetic diversity it's gonna be the same because they're fast. So I'm not sure if somebody's doing this here in the Philippines, but I haven't we haven't received any papers on how it affects the genetic diversity. So far, right now, I actually don't know if it actually affects the genetic diversity. So maybe that's a good uh topic to actually compare if there is.
O yan, may magti-thesis na on this. That's a very good thesis topic already. Yun
Yeah,
nga lang, very expensive, I guess.
yeah.
It's gonna be very expensive. So before I ask Jace for his question, let me just go back to the Q&A box. So we have here an anonymous participant who's doing a study on seasonal variations across
Mhm, mhm.
habitats. So hindi niya minention kung saan yung kanyang sampling site. So ano yung ang question is how many transects are ideal for conducting sampling in such kinds of theses and because they are planning to compare habitats and season and they want to make sure that their sampling design is reliable enough for their data collection.
Mhm. Okay, so how many whenever I do research, if I have a site, if I have one site, I would at usually have at least 5 transect lines within that site. So the usual is 200-meter long, 10-meter wide. So I hope that answered your question. If you have example, if you have 2 areas, 5 each, so you will have 10 sites. Actually, that's already a good replication for butterflies. Because chances are butterflies are flying. So so I mean there are many things to consider when working with butterflies because they fly around. So maybe in one of your sites, one butterfly is gonna be flying across all transects. And then sometimes you will find those that are uh, most specific. They just fly around their plants, and that's a good sign that there are species that are not genetic, but actually possibly specialist. So yeah, five transect lines per site. That's actually a good start.
Sige. So thanks Sir Ja. So let's go back to our audience. So uh Jace or ano ba to Antonio, Mr. Polmo, you can ask your question.
Sir, good morning. Am I audible po?
Yes.
So, good morning, everyone. I am Jesse Antonio Polmo from the Iloilo Science and Technology University. I am currently a Bachelor of Science in Biology student. So, I am intrigued, sir, about the transfer of the butterflies from Philippines to Hong Kong and from the Japanese islands to the Batanes. So, I'm curious, sir, how they can survive the storm or the typhoon, and thus the emergence of the new species of that specific butterfly can have an ecological impact or imbalance to the existing community of the certain species po.
Right. That's a very good question. How do you think those butterflies reach here in the Philippines via typhoon? So here's what we think how they actually move. They actually there's a possibility that they fly on the outskirts of the typhoon, not necessarily in the middle of the typhoon, because I mean the strong winds, something the wings of the butterfly can't survive that. So the only way possibly for them to reach is to actually fly on the outskirts of the typhoon. That one's not too strong, but it has a direction that's going somewhere. So that's what we think. We don't know. I mean, if we really want to do this kind of research, we have to attach a satellite to the butterflies. I'm not sure. I think there's probably available. That's going to be expensive. Attach it and then uh, wait until there's a typhoon and monitor how it actually flies towards the typhoon. Because based on my experience with typhoon, when I collected specimens after typhoons, some of them are actually battered. The wings are not the wings are not in good condition. They're actually it's either old or they've been through something. Because whenever I do the non-typhoon and typhoon season, before the typhoon I sampled, and after the typhoon I sampled, I noticed I recorded species that wasn't recorded the few days ago. So when I checked the specimens, it's battered. So it's possibly that they've been flying somehow in probably with typhoons. So that's what we think. We think they're probably flying through the outskirts of the typhoon. Next is, how do you think it will? This will definitely affect ecologically, but we still don't know how it will actually affect ecologically. So right now, we have a plan on doing a field work in Batanes next year. We are going to check if there will be leftovers from Japan or Taiwan butterflies there, and see. The question is, if they got there, do you think they'll establish their population? That's a good question, because some butterflies from the Philippines who reach the southern part of Taiwan, they die because the host plant are there. In this case, that butterfly from the Japanese islands went to Batanes only to find out that their host plant is there. Now the question is, do you think the adult will survive, or do you think that species will establish its population in Batanes? So if it's just one caterpillar, probably not. If it's just one caterpillar. If it's a male or a female, again, it will not survive. It will not give new generation. So it's just, maybe if the typhoons are getting frequent and frequent, then that's the time that more and more will come in. And it's just a question, maybe in the next few years, if they will establish that population. So right now, we have guesses. And right now, again, future research opportunities. Yeah, I hope that answered your question.
Ayan. Thanks, Sir Ja. Ito, bigyan natin ng puwang yung ating speaker this afternoon, no? Mukhang balak ang goy. Alam mo naman si Sir Perry, may balak to sa'yo eh. So, Sir Perry, ito yung pang Miss Universe question na tinatanong lagi sa atin ng mga scientists, ay sa mga estudyante sa atin. So, paano ka daw nagsimula? How did you begin your passion for your chosen taxon? At ano yung mga pinagdaanan mo para makarating ka kung nasaan ka ngayon? Ayan.
Yeah, I get that a lot of questions. It started during my undergrad, and then senior years. I was like, you need to decide what to do after that. And then I planned to go to med school. So I took electives na mga pre-med, only to find out na maybe med is not my field. So I guess the other option is to go to research. But I don't know what kind of research I should do. Birds, lots of bird researchers. Mammals, lots of mammal researchers. Plants, maybe. I like plants. This is why I still like host plants. Maybe plants. I don't know. So I went to the library and then tried to look for what kind of field I would like to go to. So I went to the library, and then I went to the biology books. I found three entomology books. I checked it out, and then I read, of course, scanned through some pages. And then I went back to the university after a few days. And then there was an announcement that there was a scientist visiting to have a lecture, only to find out that the lecturer was actually one of the authors of the books that I got. So I said, okay, I got this few weeks ago, and only to find out this man speaking. So during the talk, I brought the library book with me. I went to the stage and asked him to sign about the book. And yes, that book is still in Silliman Library. And it's signed, so that's very valuable. And he said, so you're really interested in working with butterflies? And I said, maybe insects, but yeah, I think butterflies a good start. And then he said, okay, let's talk about your graduate. And that's it. And after that, I went to they gave me a scholarship, went to the States, and I studied first moths of North America. And then after that, I studied butterflies of Honduras. I was in my committee, Jackie Miller. She introduced me to butterflies, and first was butterflies of Honduras, and then I worked on butterflies of Africa, Asia, and Australia, and then ended up getting interested to Asia, and then uh, ended in the Philippines. And then, taxonomically speaking, Philippine butterflies are well studied, and now I'm more interested in their distribution, host plant shifts, elevational shifts, developmental biology. This is why I study caterpillars, because I like to study their developmental stages. Maybe something we get, uh, in the field of medicine, especially in developmental biology, you will probably discover something in their development, especially the changes from the larval stages. If there are restrictions, especially in gene expressions or something like that. And then now working on mostly ecology and, you know, everything about the ecology and taxonomy about butterflies. So that's how my story, how I ended up doing this from a very I don't know, confused where to go in undergrad, med school research, and then I tried med school courses. Didn't Not wasn't for me, so I did into research. I wanted to find out. It's actually looking or finding your interest. Actually, that's the most important. That's something that you can work for many years. Something that you work that you won't get bored. That's how we usually say. A lot of students keep me asking that in class. Why are you working on this? And what makes a lot of people say you don't get bored working on this? And I would say, if you look at my computer, I probably have a lot of 10 unfinished papers from many years. And going to field exposes you to a lot of curiosity. It's like you go to the field and say, oh, why is it here? Oh, there's something new. There's something new. So, and then, of course, after going back to the field, I go to class and then actually lecture it to my class. And then that's how you, I guess that's how you, uh, what's this? Share your knowledge. And I mean, I enjoy the field work and I enjoy the teaching. So how I ended up like this is actually looking your interest and a work that you don't get bored. I hope that actually that what's this? Uh I hope you learned something of what I said.
Yeah, yeah.
Eh,
Oh, definitely, definitely. I think lahat naman tayo sa field na 'to. That's why every time I organize these lectures, actually that's why I open it to the public, even if it's just a guest lecture for my class, and then to give opportunities and, to look at scientists. We are also human, no? May bago kami nakakarating sa ganitong mga stages sa career namin, bago kami somehow nakilala sa aming mga field. We were students at first. We were also confused at first. And probably along the way, we met our mentors. And we were, as much as perhaps we were good in our field, we were also nice with each other. Kasi alam nyo, to be honest, no, if siguro hindi ko kaibigan to sila Sir Jaan nyo, tsaka sila Sir Perry, ako, ang hirap kontakin ng mga to, busy tong mga to, maraming ginagawa. Wala lang silang choice, madali lang silang i-bully. No? Wala kasi silang magagawa. Marami akong hawak, marami akong alam sa kanila eh. So hindi nila ako malabanan. But anyway, sige. So let's go to Aldrich perhaps, no? To ask his question personally. Aldrich?
Good morning po. Am I audible po?
Yes.
Alright, thank you po. Aldrich Roque, BS Biology from Bulacan State University. My main interest po kasi is macrofungi, the ascomycetes and basidiomycetes. And bigla ko lang po naisip, if you have read or done any research between fungi and Lepidopteras. For example po, in terms of spore dispersal ng mga macrofungi, have you ever seen or observed spores sa abdomen, legs, or body surfaces ng mga Lepidoptera? Or yung sa cordyceps, have you ever seen or encountered one infecting a Lepidoptera while doing research, while doing field work? Ayun lang po. Thank you po.
Definitely cordyceps. I find them randomly, cordyceps. And in terms, since you're working on fungi, there is a there is a group of moth that actually feeds on mushrooms. That's actually the bagworm moth. So they actually live or feed on mushrooms. And their pupa thrives on mushroom. I'm not sure about their pupal stages, and I'm not sure about their adults, whether they carry the spores of the fungi. That would be a good, yeah, that's a good research. If you collect fungi or mushrooms, try to check underside. If you see something like a bagworm or like it's like twigs or leaves that are attached to each other that looks like a cocoon, chances are that's a bagworm moth. So that's actually a not so documented life stages because we think they feed on plants. And then only to find out that some are actually, there are ones that are feed on lichens, the ones that are feed on ferns. And if your mushrooms will have those, definitely publish it. If you find the life history of that moth in that mushroom, chances are that's actually new. We don't know. We only found some actually some finds them on mushroom, but they actually don't publish it because maybe they just accidentally saw it. So if you find it and then maybe study if they also eat within the mushroom. Because if they pupate within the mushroom, chances are when the adults emerge, they will definitely carry the spores. So yeah, that's a good research opportunity. Good luck with that.
So thank you, Sir Ja. So I think last two questions. So apology sa ating mga
Yeah.
students, no. You can actually directly contact Sir Ja if you have some inquiry. Huwag niyo lang punuin yung inbox niya, maawa naman. Kaya hindi rin marunong mag-delete ata to ng message sa ano niya, sa email niya. But you can actually contact him personally, I guess, no, and ask for advice. At saka pag nagpapa-ID naman kayo, may preliminary ID naman sana kayo. Huwag niyo nang pahirapan si Sir Ja sa pag-ID para confirmation na lang or verification na lang, no? Yun yung mga minsan madalas nakakatanggap din ako ng mga ganyan sa estudyante nagpapa-ID, no? Pang verification lang kami, guys, kasi kailangan matutunan niyo rin yung skills ng pag-ID. Kasi kung hindi, si Sir Ja na lang ang marunong mag-ID sa Pilipinas. So you need to learn that. So last two questions from our audience. So let's first take one more question from Miss Andrea, then we'll go to Miss Maniego. Sige, Andrea?
Hello, good morning everyone, especially to you, sir. I am an undergraduate who's working on a thesis about Lepidoptera from NMSCST or Northwestern Mindanao State College of Science and Technology. And my question is, what guide did you use during identification? Can you recommend us on what did you use, sir?
Okay, what did I use before we established the Philip, which we would like to recommend that one, the Philip website, because we almost photograph, I don't know how, but most common is probably illustrated there, and some of the rare, we're still trying to find them. So how I started, aside from some of the books are very expensive and they're not available here. They're only available abroad. So what I can do is I go to before Philip, I went to the Butterflies of Singapore website, Butterflies of Borneo website, Butterflies of Hong Kong website, butterflies of Vietnam, butterflies of Indonesia, butterflies of Thailand, and butterflies of India. Chances are we have similar species, just different subspecies and maybe other rare species that can only be found in the Philippines. So I would recommend that one. And then if you go to Philip, again we have illustrations there. And then if you go to just go to Google butterfly research in the Philippines, and it will show you mga recent research in the Philippines that they will actually have illustrations of the species that you can actually use and compare in your study. I hope that's helpful.
Ayan, by the way, Sir Jaa wrote a field guide, no?
Yeah.
Naalala ko lang, no? Kasi ayaw niya i-promote yung sarili niya. Ipo-promote ko siya. Sir
That
Jaa has a field guide for Philippine butterflies. So you can actually use that as well. No?
it's available now in Shopee and Lazada. It was sold out for that's why I forgot, because it's been sold out for many months now.
Madalas siya ma-sold out eh. Yung sa'yo yung ano yan eh,
Gives you
meron tama ba
points.
series siya, di ba?
Mas cheaper siya sa Shopee and Lazada because you will have your Shopee points, Lazada points.
Paano ko
And
ipopost sa YouTube ko to? Mayroong promotion. Sige.
so yeah, you can use that. I think the one that I showed you yung nag-work sila on butterflies on different campuses in Mindanao. They actually cited my field guide there. So they used my field guide and other field guides that are available too. So yeah.
It's actually a series of field guides. Meron siya sa reptiles, meron siya sa butterflies, meron siya sa birds. But you know, it's available. I just forgot which is the publisher. So one last question for our session for today para makapag-lunch ng maaga si Sir Ja for today. So, Miss Manyeza, your question please.
Yes, sir. Good morning. Sir Ja, good morning.
Morning, good
Yeah.
morning.
Yes, my name is Manyesa Semeniego from UPB Graduate School, taking up med biology. So here's my question. Actually, light lang po ito I've been noticed that nowadays, so maganda yung topic mo about butterfly. So really caught my attractive talaga. Like now, like for business situations or tourist attraction. So this is my question, sir. Business effectively partner with researchers or conservation organizations to leverage insights from butterfly ecology. So my question is, what advisor would you give to businesses looking to incorporate ecological principles like this, inspired by organisms like butterflies, into their innovation pipelines? That's all, sir.
Okay, so innovation pipelines for I I did not get. Can you repeat again? What was it?
Sorry. Yes, Sir Ja. Ano po yung advice mo sa mga businesses that looking to incorporate these ecological principles inspired by organisms, especially these butterflies, para sa innovation pipeline nila? Yung mga techniques ba, sir? Of course, they need to preserve, protect as well, yung mga concept, di ba? Like, sa mga Singaporean or sa mga others na mga na may yung, it's business kasi may entrance yun di ba? So, advice mo sa baka others too maging ma-inspired din.
There are actually I visited some butterfly gardens, and the one that I visited was the one in Bohol, the Habitat Bohol. So she was very successful in establishing that butterfly garden. That's because again she incorporated a lot of factors. First is the species. What were the species in Bohol? What were those species feeding? Where were those species going? So she was able to set up the garden at the perfect spot where most of the butterflies converge. So he set up that butterfly garden. He planted many trees, tall trees, to provide shade. Butterflies doesn't like very hot environmental conditions. So there were tall trees and then planted host plants and actually tried to mimic how a natural forest would look like. And his and her garden is actually a tourist spot in Bohol, Habitat Bohol. So if you go there, it's a known butterfly garden. So again, in terms of innovations in the ecology, it's actually mimicking how the forest would look like, how you're going to mimic it in a way that the butterflies would actually love to thrive in that garden. Because actually right now it's gonna be difficult because there's a lot of factors in order to consider. Just like farming, just like rearing in a fish pond. It's gonna be there's a lot of factors that needs to consider in order for them to survive. I hope that answered your question.
I think you answer ano naman,
True.
a
Thank
very
you so much.
interesting answer as well. No, and again,
Mm.
with business, I think, no, I don't know if Sir Ja agree with me is the basta ang primary goal is really for conservation. I mean, of course, if there's profit and
Mhm.
that's part of it. Eh, wala tayong magagawa ron. That's part of our we need money eh. Even sa research, kailangan namin ng pera, di ba? And I think na-emphasize ni Sir Ja yun. But we really need to think about ano ba yung actual goal natin ba talaga when it comes to these kinds of things. Is it really for conservation or purely for income? So depende yun. So, once again, thank you, Sir Ja Badon, for your special lecture for my class today. Maraming salamat sa paggawa ng trabaho ko for today. Kasi klase ko talaga siya. But anyway, but before we go, perhaps some short message for our participants before we close this session for today.
Ako, sir, or
Yeah. You. Sorry.
ako, okay. Thank you for joining. I did not expect 200 plus. This was like 260 or something. Thank you for joining me, and I hope you learned something from this. And I hope maybe I will be able. I'll cite your papers uh, later on working on Lepidoptera if you are going to proceed with that field. And some of you will probably go into offices such as what's this? Department of Agriculture, Department. Maybe some of you will go to DNR. So I will see you when I'm going to apply for a permit or clearance in some of the sites. And then maybe some of you will go into the academia. And then I just had yesterday, there was a student who went to their office. He was a new student, and he said, "Sir, I actually contacted you years ago for butterflies." I'm like, really? What's your name? I said, he mentioned his name, and it's like, yeah, I remember you. So let's look at it, and then something like that. So maybe some of you will do your grad school in UPLB. So see you. And then good luck on your research to those who ask questions. So again, research is how will I call this? Research should be fun. So research should be fun when you go out into the field. Do not be upset that you're not satisfied with your data. It's probably just went on the wrong time. So what else? Depending on your interest, maybe some of you here will not proceed working with butterflies and moths. Maybe you will have your own taxa of preference. I mean, that's already a talisman to actually have your own interest on this because, again, look for a field that you will not get bored working. So if you find it, you will not have to worry about stress at work. You will be stressed at other things. You will stress. I mean, maybe Sir Eric knows that. We in academia, we have we have stress. But as much as possible, 80% of your work will have to be interesting and you will not get bored working on that field. So I hope that actually uh, um, learn something, especially those who are still who are still confused. I mean, you're still undergrad, you're open to many doors. Where are you gonna go? If it's not there, you go to the other door. If it's not this field, go to the other field. I'm already I'm turning 36 this year, so my door's already established. But maybe go into explore that door already. So again, you guys are still young, and many doors are gonna be opening for you, many opportunities, and maybe take all of those and then choose which one you will definitely enjoy the most. Okay, I hope
Yeah, thank
that's
you.
something.
Thank you, Sir Ja. Actually, my original plan was to fly you in, no? Because we have some students who would like to study some butterflies in the UPB campus. Oh, don't worry, pag budget is available already, we're gonna fly you in soon, no?
Yeah,
So that you can
mag-low
see
gas
the
prices
UPB
din.
campus.
Yeah,
Oo.
actually
Sobrang
mag-low.
mahal kasi. So sabi ko, ano na lang, i-online na lang muna natin si Sir Ja. But definitely, we will invite you to come to UPB for a collaboration. So our students hopefully are excited for that, no? To see you in person. Single pa ata si Sir Ja, hindi ako sure, no? Pero literally gawin to. So ingat-ingat kayo. No? So, sige. So once again, thank you everyone for joining us today. There is also another one this afternoon on Anne. So
Very
for Sir Perry. Benavendes. I hope you can join us again this afternoon. So, once again, Sir Ja, thank you very much. I hope we have inspired our students, no, to
right.
pursue arthropod research. Kasi very few talaga tayo in the Philippines who are doing arthropod research. So, once again, everyone, so I'm gonna end the meeting. Maraming salamat ulit, Sir Ja. Yung
Thank
mga
you.
pabuenas sa'yo, tsaka ako na pag ako'y nasa Luzon. So, maraming salamat.
Thank you. Thank you.
Thank you, everyone. Thank you, everyone. Sige, maraming salamat. Salamat, sir. Maraming salamat sa lahat.